If you fly from Auckland to Wellington on a nice day, you'll see Taranaki, that's the name of the mountain and the province where I grew up. That's New Plymouth, my hometown, Pukekura Park in the middle of town. Follow the river up from there, you get to the Boulder Brooklands. We try to get up there every year to catch up with the old gang at the WOMAD Festival. Keep going in that direction and you get to the Tianui stream. We live just on the other side of that stream at the outskirts of town. There's the old swimming hole where I spent many summer afternoons as a kid. Our house is just up the top of the hill behind that. And if you follow the Tianui stream back down into town, you get to my old high school, which is where you would have found me in 1969. I was the youngest, smartest and best looking of three boys. My dad was involved in horticulture one way or another all of his life. During the time that I was at high school, he built up a successful business growing flowers and exporting flowers to Japan. My mother was a school music teacher. Despite living in the North Island, Mum and Dad are both mainlanders. Mum's from Lumsden in Southland, where her dad was the postmaster. Dad grew up on an orchard in Clyde at a time when apparently there were no cameras. But there used to be cameras, because there's my gran with her parents, my great-grandparents, in front of the pub that they used to own in Clyde, which is still there. And they owned the other pub on the corner, which is also still there. Dad's other grandfather, my great-grandfather, lived in Dunedin, worked at the University of Otago in the School of Mines. Most famously, he once went on a 6-month field trip in South Westland and came back 3 years later. There's Helen and the girls up at Paulin Falls, summer before last. There's me and Sam, wondering if we can get to the base of the falls. Answer, not if you want to be home for dinner. And there's my great-grandparents, Robert and Catherine Paulin, at her parents' place, my great-great-grandparents, and that house is in Kaikoura Valley, just a few kilometres over in that direction, and it's still there. But I wanted to come back to 1969, which was a very auspicious year for me. Out of about 30 boys in my Year 9 class came 3 PhD scientists, 2 All Blacks, and 1 serial killer. But there's one occasion that really sticks in my memory from that year. It was during a Latin class one Monday morning in the middle of winter. The Latin master, who we called Fish, I don't know why we called him Fish, suddenly strode to the back of the room and he picked up this kid by the scruff of the neck and he stood him up and he said, "What are you doing, boy?" At that point, we saw what Fish had seen. This kid had an earpiece in. He was listening to his transistor radio in Latin class. This is definitely a caning offence. So there's a moment's silence. And the boy, whose name was Brett, said, "I'm listening to the moon landing, sir." And there was another moment's silence. And Fish said, "We'll take that earpiece out so we can all listen to it then." So we all crowded around this transistor radio and listened to a live radio broadcast from the moon. I am a little bit difficult to make out in this photograph, but I can verify that's definitely me in the background. To be honest, I don't remember anything of what we heard that day. I just remember the drama of the occasion. These guys are in a metal box, in a vacuum, in zero gravity. They've got to steer this thing and land it on the moon. There's just a million things that could go wrong. They could spin out of control, they fly off into space and are never seen again, they crash into the moon and they're all killed. They tip over when they land and they can't take off again. In the event, it all went well. And this was a big deal in 1969, especially for a 12-year-old, because I was a Sputnik baby. 12 years earlier, the Soviets had launched the first artificial satellite into orbit. And on this day, we knew that the good guys were back in front. So obviously at the time, my ambition was to be a rocket scientist, but at some point in the following 5 years, I read an article about DNA sequencing that explained how DNA sequencing was going to answer all questions in biology and cure all diseases. So I thought, yeah, I'll sign up for that. So I enrolled at Otago University to study biochemistry. I didn't enjoy first year biology very much. Actually, our lectures were in the old zoology lecture theatre, which is where the tea room is now. So if you're having a cup of tea, you'd be suspended in the middle of a large lecture theatre which used to occupy the back part of that building. I tended to get distracted by the computer centre, which was between Unicol and the zoology department, And so I ended up hanging out with other nerds and learning how to program. Of course, that was on a mainframe computer the size of a bus. There were no personal computers, no internet in those days. We didn't even have a television set in the flat. If we wanted to watch Monty Python on a Monday night, we'd have to go down to the cafe at the Student Union. So we had to make our own entertainment. Here we are at our flat in St. David Street. There's Unicol in the background. Having a wild and crazy time during orientation week, putting in a vegetable garden. Now, even if you know this person on the left, you may not recognize her in this old photograph. But if you ask yourself, who turns up to plant lettuces carrying a saw, then if you do know her, you will recognize immediately that it is, of course, the absolutely fabulous Helen Davies. In the summer of 1976, Helen and I went to Australia. And in the week before Christmas, we camped in the Parachilna Gorge, which is in the Flinders Ranges, north of Adelaide. At the eastern end of the Parachilna Gorge is a little town called Blinman, where we stopped for a beer at the pub. This is another one of those days that sticks vividly in my memory, because when we went into the pub, there was a bunch of Aussie guys in there, in their work gear, talking very excitedly about science. If you're a Monty Python fan, you'll know why this was so memorable. Here we are in a pub in the Australian outback, witnessing a live replay of the famous philosopher's sketch from Monty Python.
Hello, Bruce. How are you, Bruce? Gentlemen, I'd like to introduce a chap from Pommyland who'll be joining us this year here in the philosophy department at the University of Wollongong. Michael Baldwin, this is Bruce. Michael Baldwin, this is Bruce. Michael Baldwin, this is Bruce.
Is your name not Bruce, then?
No, it's Michael.
That's going to cause a little confusion.
So if you could go back and tell the 20-year-old Mike Paullin that he'd be back decades later with the same girl, still driving a Holden, he might have thought that sounded plausible. But if you told him that the reason you came back was because you thought that the secret key to understanding how brains work was hidden under a rock behind the North Blinman pub, I don't think he would believe that. But let's have a look at what's behind the North Blinman pub. There's clearly something going on with these rocks. In fact, what we're looking at here is an ancient seafloor that's been a bit scrunched up. If we zoom down on a relatively unscrunched up bit of it, we get to the Brakina Gorge. So the Parachilna Gorge is up here with Blinman over on the right. We're about 25 kilometers south of there. What you're looking at here is that at some point in the past, the continental plate has snapped and the western side of it has been pushed up over the eastern side of it, much like what's happening in Kaikoura right now. So this is the corner of an ancient broken piece of Australia that used to be sticking up a lot more. It's been worn down, and what's left of it is the Flinders Ranges. And what makes this particular chunk of continental plate so interesting is that over here on the left, the surface here, is a shallow seafloor that's dated to the early Cambrian. And if you're driving through the gorge, you actually get to a sign at this point marking the boundary of the lower Cambrian, and at that point you can get out of your car and you can see animal fossils in the rocks. Over on the right here, this is tillite, so this is evidence of glaciation. This is the tail end of the Cryogenian period. Across here, we've got 8 kilometers, which contains sediments laid down in a shallow ocean over a period of about 100 million years, including part of the lower Cambrian. Now, this is really interesting for zoologists because when Darwin wrote his famous book in 1859, he pointed out that if you dig down through the rocks, you find animal fossils, and the deeper you dig, you find animals all the way down until you get to a certain point where there's no more animals. And it was a problem that Darwin knew about, and he pointed out that at the point where the animals disappear from the fossil record, we find most of the major different groups of animals that we see today. And this is a problem for Darwin because, of course, his theory is that animals evolve by gradually modifying earlier animals, And so he said there must be some fossils somewhere that we haven't found, or there's some explanation as to why the animals just suddenly disappear from the fossil record, giving the impression that they just appear suddenly at some point in the past. It was another 100 years until the 1950s until paleontologists started discovering animal fossils in this region of the Flinders Ranges and in other places around the world. If you go there now, you'll find at the other end of the gorge there's a marker next to a stream marking the beginning of a geological period called the Ediacaran period. So that's named after the Aboriginal name for this area, the Ediacara Hills. I think Helen and I can be excused for not noticing this at the time. If you look at the date on that marker, it's 2004. The Ediacaran period has only been a thing for less than 20 years. It's only occurred to me in the last few days while I was preparing this talk that in the 1970s this place was crawling with paleontologists. If you were there on a hot summer's afternoon in 1976, just before Christmas, you shouldn't be surprised to find a bunch of them in their work gear in the nearest pub, which is in Blinman, and you shouldn't be surprised if they are talking enthusiastically about science and the implications of science, because this is one of the most important scientific discoveries of the 20th century, and these guys were in the thick of it. Before we leave Burkina Gorge, we'll just take a look at what was there in the middle of the Ediacaran period. On land, there's nothing. There's no multicellular organisms, no animals or plants on land. It's a desert. The animals and plants are in the ocean. We dive down and have a look. There's a lot of stuff, but one of the most common fossils is this thing called Dickinsonia, this very flat animal that lived on the floor of shallow oceans at that time. Luckily for us, because there's nothing on land, every time there's a major storm, these things get buried in silt and preserved in pretty good detail. Entire ecosystems get preserved in detail. So here's a body fossil, and here's the trace fossils out behind it on an actual slab of rock. That's the actual slab of rock that fossil came out of. There it is. There's the body fossil sitting there with the trace fossils behind it. So this one slab of rock was the basis of Felicity Coote's PhD thesis. Actually, one of her thesis advisors is Corey Bradshaw, who used to be a PhD student in this department. This one slab of rock has over 100 animal fossils on it, so there's an awful lot of fossils in those hills. Of course, Helen and I were completely oblivious to this at the time. I ended up with a degree in mathematics. Here's the academic staff and graduate students in my final year at Otago. About this time, Apple Computer got started, and the very first Apple microcomputer that came into New Zealand was bought by the zoology department at the University of Auckland. So these are fantastic machines, with the one downside that they came with absolutely no software, right? And you could play Pong, or you could program these things in BASIC. That was it. So they advertised for a programmer, and I applied for that job. I knew how to program, and I even had a little bit of biology in my degree. My job was to help the academics and students with data analysis and computer modeling, which required me to learn something about data analysis and computer modeling. One of the people I worked with was a neurophysiologist called John Montgomery. His research topic was the cerebellum of sharks. So John explained to me that in humans, the cerebellum is responsible for fine control of movements. So the classic test for cerebellar dysfunction is this finger-to-nose test. So your doctor might ask you to move your index finger rapidly backwards and forwards between the tip of your nose and the tip of your other index finger. If your cerebellum's damaged, you'll tend to poke yourself in the eye. You'll miss at the other end. Cerebellar patients tend to be kind of awkward and unsteady. If they close their eyes, they might fall over. But in sharks, the cerebellum, or at least a large part of the cerebellum, is involved in something else. It's tied in with the electric sense, the sense that allows sharks to detect other animals by detecting the electric fields around them, and the lateral line sense that allows sharks to track other animals by sensing movements and sense other things around them by sensing water pressure and water movements, and also in processing information from the vestibular system. So in sharks, the cerebellum seems to be involved in sensory processing, in tracking the movements of things around the animal and in tracking the movements of the animal rather than in controlling the movements of the animal as it is in humans. So I said, well, I think I can explain what's going on there, because this finger-to-nose test, that's the moon landing problem, right? You've got to figure out what muscles to contract in order to control this movement without flying off into space, without crashing into something, without spinning out of control and falling over. And what I didn't know when I was 12 years old, but I did by this time, was that the pilots of the lunar lander didn't actually directly control these thrusters. That was done by the Apollo guidance computer. So there's an onboard computer, and what the pilots have to do is tell the computer where they want the vehicle to go, and the computer calculates how to activate the controls to make the vehicle do that. This is called feedback control. What the computer does is it looks at the difference between the state that the vehicle's in and the state that the pilot wants the vehicle to be in, and then it does some algebra and calculates how to activate the thrusters in order to move the vehicle towards the state that the pilot wants it to be in. So it's very simple in principle, but there is a little problem with it, which is how does the computer know what state the vehicle is in, right? So this is the orientation of the vehicle and whether it's tipped or rolling or whatever. The solution is that you replace the unknown true state of the vehicle with a statistical estimate of the state of the vehicle obtained from sensor data, principally radar and gyroscope data. This was figured out in 1960 by a guy called Rudy Kalman. His solution is called the Kalman filter, and he showed that if you replace the true state of the vehicle with the estimate of the state of the vehicle obtained from a Kalman filter, then it all just works. Now, you know, when I'm giving a talk, you're not getting out of here without seeing some equations. So there's some equations. These are the Kalman filter equations. This is the algorithm that was running on the Apollo guidance computer in the lunar lander. This algorithm runs in aeroplanes, in robots and guided missiles and all kinds of things today. It has all kinds of other applications. This algorithm is what makes it possible for human beings to fly jumbo jets. So this is what made it possible for my dad to pick flowers in New Plymouth on a Saturday and have them in the markets in Japan on a Monday morning. I suspect that Rudy Kalman is the most important human being you've never heard of. So John Montgomery said, well, that sounds fairly interesting. You could turn that into a PhD. So I went away, did my OE, came back and enrolled for a PhD under John's supervision. The bottom line of my PhD was that the cerebellum is a neural analog of a Kalman filter. So the Kalman filter is a model of what the cerebellum does. And the implication of this theory is that the function of the cerebellum hasn't changed over the evolutionary history of the vertebrates. It is essential for fine movement control, but that's because it provides the motor control computers with the state information that they need in order to control movements. So what's happened in evolution is simply that the vertebrates, or us, the land vertebrates, have lost those senses, the electric sense and the lateral line sense, that only work underwater, and that just makes it less obvious that the specific role of the cerebellum in fine movement control is state estimation. And before I leave this slide, just mention that those senses, the electric sense and the lateral line sense, are kind of developmentally related to our auditory sense, our sense of hearing, but they're functionally different from our sense of hearing. They are most similar to our sense of touch. So we tend to call them touch at a distance. So when a shark is sensing something using its electric field or using its hydrodynamic sense, it's as if we could feel something before we touch it. We can feel something that's away at a distance. So we call those senses touch at a distance. Also, as part of my PhD, I did some neural modeling. At the time, the cerebellar circuit was pretty well understood, or we thought it was pretty well understood. There was this classic book that had been written going through the detailed circuitry of cerebellum, and it finished off by saying that we pretty much know everything about what's in the cerebellum, and so it's only a matter of time for the mathematicians and the computer scientists to figure out how the circuit works and how it controls movements. I actually found that it was pretty easy to model this circuit and show that it could function as a Kalman filter. The problem was that it was also pretty easy for me to model the circuit and show that it could function as something else. And if you gave me another similarly complicated circuit, I could show how that other circuit could operate as a Kalman filter. So the problem is that we can model this, we can model everything, we can model the cerebellum, we can model the neurons, but we're clearly not explaining anything. And I'll just address the elephant on the slide. This has had to remind me of, there's a famous saying in numerical analysis, which is, give me 4 free parameters and I'll draw an elephant, give me 5 and I'll wag its tail, which is making the point that if you have enough degrees of freedom, if a model has enough degrees of freedom in the model, they can make that model do pretty much whatever they want. And so that's the problem here. There's so many degrees of freedom in what we know about this neural circuit that anyone can make it do anything you want. When I finished my PhD, I went to Los Angeles. I did a postdoc with Dennis O'Leary at USC on vestibular system, neurophysiology and computer modeling and data analysis. Then I did another postdoc with Jim Bauer at Caltech. So Caltech just had a new program at that stage called Computation and Neural Systems. This was the heyday of Sun workstations and Silicon graphics workstations, and it was becoming possible to do really spectacular things with desktop computers. And Jim literally wrote the book on detailed, realistic biophysical models of neurons. Jim's approach was to constrain neural models by adding fine detail down to the molecular level. I was always a bit skeptical of that approach because it seemed to me that when you're adding those microscopic details into a model, you're not just constraining the model, you're also adding degrees of freedom, so you're not getting rid of that elephant tail wagging problem. Around that time, I saw an advertisement for a job for a comparative neuroscientist in zoology at the University of Otago. And so I applied for that job, and here I am. When I became an independent researcher, I started a collaboration with Larry Hoffman, who I'd met in Los Angeles. He's an anatomist and an electrophysiologist. I prefer to work on the modeling, data analysis side of things, so that collaboration worked well. It's still working well. And instead of starting at the bottom with the molecular details of neurons and trying to build up a picture of how neurons function, we started at the other end. So we looked at neurons from the point of view of information theory. We modeled them as communication devices. So we're asking, what information are these neurons transmitting to the brain? Which is, of course, a key question in understanding what happens in the brain. It took us a while to get there, but we came up with this model that can describe the complex behavior of vestibular sensory neurons in a very simple way using only 2 free parameters. So I'm very happy about this because there's no tail wagging going on here. But when you stand back and ask, what is this telling us about information transmission in the vestibular nerve? And what it tells us is that these neurons are about as inefficient as they could possibly be in transmitting information from the receptors into the brain. And when we dig a little bit deeper into that, it looks like what's going on is that at the receptor level, at the molecular level in the sense organ, the molecules that detect movement are so sensitive to movement that they detect their own thermal noise vibrations. And so those thermal noise vibrations are being picked up, encoded in the spike trains of the neurons, and transmitted to the brain. And that doesn't make any sense at all. It's very energetically expensive to transmit information using spiking neurons, so why on earth would you waste all this energy transmitting noise to the brain? Noise, by definition, doesn't carry information. So we'd come up with this beautiful model, but it just produced more questions than answers. The answers came in a surprising and kind of roundabout way. In 1999, I got a call from an old friend and colleague at Caltech, Chris Assad here, who was then working at the Jet Propulsion Lab in Pasadena, the headquarters of NASA's unmanned space program. He still works there. He said that NASA had had a couple of high-profile disasters recently. In particular, the Sojourner rover had trundled down its ramp off the lander on Mars and promptly got stuck against a rock. And it's a little embarrassing when you've spent hundreds of millions of dollars to get that thing up there. So they're looking at rebooting their robotics program, building, smaller, cheaper, smarter robots, more robust robots. And so one of the ideas is to talk to biologists about animals and animal design. And for example, you never see a cockroach run down a ramp and get stuck against a rock. This never happens. So Chris invited me to join the biological computing group, and I spent 5 or 6 summers, or Dunedin winters, I spent in the California summer working with the biological computing group at NASA JPL. This is my dream job, right? Sputnik baby, moon landing boy. NASA's paying me to build robots and play in the sandpit with my friends and let them know if we find anything interesting. Which we did, but that's a story for another day. But I do want to mention one thing that spun out of this. Some of you are aware that I do this kind of animal-inspired robot program in schools and science fairs and things, where the kids make their own simple robots and then they play with the robots with their friends, and then they get to keep the robots and take them home at the end of the day. Parents and teachers sometimes say, well, this is cool. Where did you get the idea? And I say, well, just by playing with robots in a sandpit with my friends, because you can learn an awful lot about biology and engineering and maths and physics and coding and design just by making your own simple little robots and playing with your friends. While I was hanging out with biologists and engineers, I learned a few things. In particular, I learned something really interesting about the Kalman filter. For the next minute or two, I'm going to be talking to the statisticians in the audience, if there are any. It's going to get slightly technical, but hopefully the rest of you will understand where I'm going with it at the end of that. So here's the Kalman filter equations, and here we are calculating the best estimate of the vehicle state. We're also calculating this other thing, this thing called a covariance matrix. It's no surprise to statisticians that you need this other thing in order to calculate the best estimate. But if you stand back and look at that box and say, what's being calculated in that box, that's a probability distribution. The Kalman filter is not just calculating the best estimate of the vehicle state, it's calculating the distribution of all possible states that the vehicle could be in with the associated probability of being in any of those states. And you might suspect, and you'd be right, that that probability distribution is the Bayesian posterior distribution given the available sense data. So what this means is that the Kalman filter is Bayesian. So I know what you're thinking. If you're not a statistician, and actually probably even if you are a statistician, you're thinking, why do I care about being Bayesian? Well, you should care about being Bayesian because evolution gave your ancestors a choice. You can be Bayesian or you can be dead. You might think that if you're a zebra and you see a lion approaching, it would be a good idea to run away. But here's some zebras, and they can see the lion approaching, but they're not running away. What's that about? The answer is that if you run away at the first sight of a predator, you spend a lot of energy running away that might be better spent doing something else. For example, at this point, your best option might be to gather more information about the predator, and that seems to be what the zebras are doing at this point. Over evolutionary time, if you run away too soon, you're just as dead as if you run away too late. So you might prefer not to be eaten today, which is perfectly understandable. But if you run away too soon and some other zebras eat a little bit more grass before they run away, over evolutionary time, they will outcompete you. You have to make your decision within milliseconds, but evolution is playing the long game. So this is just one example of a decision that all organisms, from bacteria to professors, have to make all the time. What's the best thing I could be doing with my time and energy right now? And the answer is, it depends. It depends on what the state of the world is right now and what's going to happen in the future. And you have the same problem that the Apollo guidance computer had, which is that you don't know the true state of the world and you don't know what's actually going to happen in the future. Your best option is to make decisions based on what you know about the world and a model of what you know about how the world works. And that's Bayesian decision theory in a nutshell. Make the best decisions given what you know and what you want to achieve. An organism doesn't actually have to do those Bayesian calculations to make the right decision or to make a good Bayesian decision. For example, a bacterium has to decide what genes to express, what enzymes to express in a given environment. Natural selection takes care of that automatically. A bacterium that correctly hooks up its receptor molecules to gene expression in such a way as to most efficiently digest whatever nutrients are available in the environment is going to be selected over other bacteria. Over the long term, the only way to beat a Bayesian is to be a Bayesian with more information. Especially in situations like this, where there are very high costs associated with making bad decisions, there's going to be strong selection pressure to gather more information. There's going to be selection pressure to build models and to be able to do these computations in real time, rather than waiting for natural selection to figure this out over multiple generations and tell you what the right answer is. So organisms can make good decisions. They can behave as if they are doing Bayesian probability calculations without actually doing those Bayesian probability calculations. But in certain situations, there's going to be an advantage in actually doing those calculations if they can be done quickly and cheaply. And this is one such situation where there's an advantage in being able to make decisions on a millisecond timescale using information that's available at the time the decision has to be made. Another thing I learned by talking to engineers and evolutionary biologists at JPL is that if we want to launch a robot invasion of Mars, then robots that mimic big cats or dogs or zebras or humans are a terrible idea. When animals invaded Earth, the way for vertebrate animals like us was laid down by simpler animals with simpler nervous systems. By the time we vertebrates showed up, the infrastructure was in place and the world was ready for animals like us. This reminded me of something that I'd noticed when I did my PhD. Human brains are just giant fish brains. When I started my PhD, I had the idea that shark brains are much simpler than human brains. But it isn't true. Shark brains are not simpler than human brains, they're just smaller. If anything, our brains are simpler because we lost some major sensory capabilities, those touch at a distance sensors, when we invaded land. So this got me thinking about animals that preceded the vertebrates and what their nervous systems looked like. Eventually, I decided that the key question was not who were the first animals with nervous systems, but who were the last animals without nervous systems. Right around that time, geologists were hammering the Ediacaran marker pen into the side of the Burkina Gorge, and I started thinking about what's under those rocks behind the Blenman pub. And that takes us back to this animal Dickinsonia in the Ediacaran period. So these are very flat animals that are gliding along the seafloor, pausing intermittently to dissolve away these microbial mats, these bacteria and single-celled eukaryotes that are forming the slime on the seafloor. So this is the very first fossil evidence we find of animals moving, and this represents the introduction of a new metazoan feeding mode about 555 million years ago. So that's microbial mat grazing, motility with external digestion. So these things are moving and they are dissolving things, digesting things outside their bodies and then taking up the nutrients. This happens within a few million years of the first evidence of carnivores and of the first evidence of animals with nervous systems. So let's take a look at how we think these animals are related to us. At the beginning of my talk, I talked about my whakapapa back about 4 generations. I could have kept going to 17th century France, but at a certain point it's easier to go to the other end and start working backwards. So this is my family tree, and obviously your family tree also, from the time that we separated from the choanoflagellates, which on current evidence is sometime in the Cryogenian period, maybe 800 million years ago. There's a lot of information on this slide. We could spend an entire semester in a graduate course discussing questions that are raised by this phylogeny that I've drawn here. But today I just want to use it to tell you a couple of fun facts about our relatives. So first of all, notice that this phylogenetic tree includes modern animals, includes groups of modern animals, including us, the chordates here, and it includes this extinct group of Ediacaran animals, the petalonamids, which includes Dickinsonia. The first fun fact is which modern animal is most closely related to this extinct Ediacaran mat grazer, Dickinsonia. And the answer appears to be placozoans, a group of animals that you've probably never heard of. The second fun fact is that with modern genetic tools, we can ask questions like, which other animals, apart from animals in our own group here, the deuterostomes, has brain chemistry that's most like human brain chemistry? And just in case there's somebody listening who knows something about brain chemistry, what I mean by that is neuropeptide expression. Now, you might be rooting for some kind of clever insect or a squid or an octopus or something over here, but the answer is here, the placozoans. That might be a bit of a surprise if you've never heard of placozoans, but it's probably even more of a surprise if you have heard of placozoans, because placozoans are animals that don't have nervous systems. They don't have neurons or nervous systems or brains at all. So the animal whose brain chemistry most resembles human brain chemistry outside of our own group is an animal that doesn't have a brain. If you don't know what a placozoan looks like, now you do. This guy's about 100 microns across, looks like a giant marine amoeba, but is actually a multicellular animal crawling around on a microscope slide using cilia. These things are very flat. They're basically two cell layers, like a collapsed epithelial ball. I could look at these things for hours, mainly because me and my students have spent hundreds of hours looking down microscopes, not finding these things. But let's move on. This is trichoplax feeding on a kind of picoalgae called Rhodomonas. That's the pink things. It's releasing its digestive juices underneath it, breaking up the Rhodomonas cells and drinking them like soup. The pink clouds are the red pigment released when the Rhodomonas cells break up, and you see those pink clouds fading away as the trichoplax digests it. Note that this video is sped up. This is about 30 times faster than a real trichoplax. What I think is really interesting here is that trichoplax is not just spritzing out digestive juices all over the place and sweeping up the remains of its prey like a microscopic combine harvester. It appears to be locating and targeting small clusters of prey underneath it. Evidently, it can taste things on its ventral epithelium and only release its digestive juices when it tastes something worth eating. That makes sense because those digestive juices are expensive. With this new feeding mode, once you spray your digestive enzymes out into the world, you never get them back. So you'd better check that there's something underneath you worth eating and only release enough juice to digest what you can taste. So trichoplax's digestive epithelium, its stomach, seems to map the distribution of nutrients underneath it and make good decisions for efficient foraging. Now, trichoplax obviously doesn't have a brain. Or does it? Because what is a brain? Our brains are sheets of epithelium containing maps of relevant aspects of the world with mechanisms that make decisions and control actions based on those maps. And trichoplax has that. The only difference is that trichoplax is a much simpler animal in a much simpler niche. It has a correspondingly much simpler map, which is on its stomach in direct contact with the part of the world that it maps, rather than folded up inside a hard shell like our brains. I say on the slide that trichoplax has a map of the world on its gut, but it doesn't really have a gut. A gut is a tube with digestive epithelium on the inside. Guts didn't evolve for another few million years after Dickinsonians evolved, when some animals first rolled their digestive epithelium into a tube and fed by shoving food in at one end. There's good reasons why Ediacaran animals might want to do that, but that's a story for another day. For now, I just want to point out that if some animal like Dickinsonia wants to evolve guts, then they have a problem. If their ventral epithelium is not just doing the digestion, but also doing the mapping and decision-making, then if they form a gut, if they roll their digestive epithelium into a tube so that it's no longer in contact with the surface of the world that they live on, then they'll have to set aside some part of the digestive epithelium to keep making the maps and keep making the decisions. The set-aside region of the digestive epithelium could specialize in doing the mapping and decision-making and let the gut do the digesting. In case you missed the point, another way of saying mapping and decision-making is perception and cognition. So the set-aside region would be an epithelium whose role is to decide what to do based on what it perceives. That's what we call a brain. This scenario suggests that brains may have evolved as a consequence of selection pressure for gut evolution. But I'm going to suggest another scenario. I'm going to show you a model that explains why and how Ediacaran mat grazers could have set aside part of their ventral epithelium to specialize in perceiving the world beyond the edge of their body and making decisions based on what they perceive, independent of any selection pressure to evolve guts. So let's take a closer look at this digestive epithelium and how it works. The feeding mechanism of trichoplax and the presumed feeding mechanism of Dickinsonia involves releasing acid and digestive enzymes into a space underneath the animal. This allows these animals to digest large numbers of eukaryote cells simultaneously. It's way more efficient than the feeding mode of their Choanozoan ancestors who ate bacteria individually by phagocytosis. Because Dickinsoniids are very flat, it would be easy for one such animal to glide over the top of another and start digesting it. So this new feeding mode, which evolved to digest microbial mats, would have made it possible for Dickinsonia to eat each other. As I pointed out earlier, being able to move and eat each other is a big deal for animals. It creates huge selection pressure for being Bayesian. So that's why I'm so interested in Dickinsonia. They represent a watershed moment in our family history. The instant that our ancestors first started to crawl over and eat microbial mats, they arrived at the top of a slippery slope leading to the Bayesian brain. But how did that happen? We have acid being released across the ventral epithelium. Acid is hydrogen ions or protons, so there's a proton pump here pumping positive ions across the digestive epithelium in this direction. There's negative bicarbonate ions flowing in the other direction. This means that there is an electric current flowing vertically down through the animal. That's going to create an electric field whose strength falls away with distance from the animal in every direction. That's fairly interesting because I did a PhD on shark electric sense. And I know that all animals in seawater today, including fish and crabs and snails, and even you and me when we go for a swim, are surrounded by an electric field generated by ion flow in our guts. This is how sharks see you up close and can bite you even though their eyes are pointing sideways. If there were sharks in the Ediacaran period, they could have hunted Dickinsonia. Of course, sharks were not around for another couple of 100 million years. But the point is made that if an animal in seawater is surrounded by an electric field that gets stronger as you approach from any direction, then that animal is going to be easy prey for a carnivore with an electric sense. On the other hand, having an electric sense would make it possible for a grazing prey animal to spot another animal approaching and run away before it makes contact. This is that touch at a distance thing. When there are carnivores about, there's selection pressure on animals to feel other animals coming towards them before they touch. So we have to ask the question, did Dickinsonia or some Dickinsonia-like Ediacaran animal have an electric sense? We may never know whether Dickinsonia had an electric sense, but trichoplax does. Here's a cross-sectional diagram of trichoplax. The digestive epithelium is in the central part of the ventral side. These pink cells are so-called gland cells that release the acid and enzymes. Around the outer edge of the ventral epithelium, in a zone around the digestive region, there are these cells that I've colored green. These cells have electric field sensing molecules in the apical face, in the part of the cell membrane that faces out into the world. Those electric field sensing molecules are voltage-gated calcium channels, resembling the voltage-gated channels in shark electroreceptors. At the other end of these cells, there are vesicle release mechanisms, the kind of mechanisms that you would normally find at a synapse in an animal with a brain, indicating that these cells are releasing some kind of neurotransmitter or neuromodulator, even though they don't have neurons to transmit to or to modulate. We don't know what that chemical is in trichoplax, but in sharks and shark electroreceptors, we know that the neurotransmitter that gets released is glutamate, which I think is really interesting because glutamate, of course, is the delicious savory taste of meat. It's umami. It's that taste of miso soup. Trichoplax are, in fact, attracted to glutamate as we are. So you can think of electroreceptors as translating electric fields, which are cues for an animal at a distance, into a taste cue. In other words, they give trichoplax a taste at a distance sense. And that cue could be used to, for example, trigger an escape behavior. So in a prey animal, in a grazing animal, if it detects an electric field, it can use that as a cue to trigger some escape behavior. The escape behavior in trichoplax is to just suddenly roll up into a little ball and float away and never be seen again. I've lost a few of them that way. The problem here is quite simple. Trichoplax just has to figure out how far away an approaching animal is and run away if it gets too close. Since the approaching animal is surrounded by an electric field whose strength depends on distance, the decision problem comes down to setting a threshold for electric field strength, and if the electric field gets stronger than that, you trigger the escape behavior. As I pointed out, natural selection will take care of that. If trichoplaques can set the threshold for their escape behavior in some heritable way, natural selection will select the ones that make the right decision. But as I also pointed out, there's an advantage if trichoplaques can make this decision in a Bayesian way. In other words, if it can take account of the uncertainty in its estimates of how far away the predator is when it makes that decision. Remember that the problem here is that the ideal decision depends on how far away the predator is. But you don't know that. You have to make the decision based on what you know about how far away the predator is. So the first step in making a Bayesian decision about when to escape is to calculate the probability distribution of distance to the predator given the data that you have. So let's look at the data that's available to the placozoan. That's the states of these electroreceptor molecules. Don't worry about the equations. The bottom line is in the graph. The blue line is the strength of the electric field at a given distance, and the red line is the probability that a receptor molecule is in its open state if the field is that strong. The probability of being in the open state increases as the predator gets closer. Over on the right, there's about a 15% probability of being open when there's no external field. That's the effect of thermal noise, which flips the channels open at random, even when there's no predator. That random noise and sense data is just the price of sensitivity. If we had to wait until our receptors agree that we're about to be eaten, we'd be about to be eaten. Next, I'm going to show you a simulation of a placozoan model computing the probability distribution of predator location using this noisy data. You'll see a placozoan in the middle of the screen and a predator approaching it. The prey has a ring of electroreceptors around it that flash bright yellow when they open. The grey bit in the middle is the digestive epithelium, which contains the taste map of the world under the prey. In the marginal zone around the edge of the animal, I've computed the probability distribution of predator position from the receptor states. That's a taste at a distance map. Bayesians call that probability distribution the observer's belief about predator location. I've projected the prey's belief into the world around it so you can visualize its perception of predator location. This is all done using standard numerical Bayesian methods. I can do that because I have a brain and I can program a computer. The question is, how could an animal like trichoplaques do it? When the simulation runs, you'll see another panel representing a placozoan chemical brain model. The yellow dots are sensory molecules. They're released when the receptors are open and they diffuse into the marginal zone. The pinky red dots are belief molecules that diffuse randomly in the marginal zone. When a sensory molecule collides with a belief molecule, it catalyzes the formation of a new belief molecule. There's an image of the sensory molecules and belief molecules projected into the outside world, so you can visualize what they're saying about the location of the predator. Here's the simulation in its initial state. You can see that at this point the numerical Bayesian in the middle thinks that there is a predator out there somewhere, but it's very uncertain about where it is. Similarly, the beliefs of the chemical Bayesian on the left are scattered all over the place. When the simulation starts, the predator approaches, and the Bayesian probability distribution converges onto the leading edge of the predator. Over there on the left, the belief molecules concentrate around the leading edge of the predator in a similar way. The distribution of belief molecules turns out to be pretty close to the Bayesian probability distribution. It's an internal image of the approaching predator that's almost as good as the best picture that it's possible to get using this sensory information. Of course, evolution couldn't care less about little pictures in your head. It cares about you making good decisions, and this picture is just a means to an end. When there's no uncertainty about predator location, the best decision is simply to run away when the predator gets too close. When there is uncertainty, the best decision is to run away when there's a certain probability that the predator is too close. Here's the probability that the predator is closer than 50 microns over multiple trials. Blue's the numerical Bayesian calculation and brown's the chemical brain model. It's very easy to compute that probability from the chemical brain model. Just count how many belief molecules are in the region of the map representing the region of the world within 50 microns. This is one small step for a man with a computer, but a giant leap for a placozoan without a brain. How could a placozoan do this calculation? This picture shows a cell in the map. I call that cell an M cell after a neuroscientist called Ludwig Malvner for reasons that we don't have time to get into, but you can look that up. The region in the world corresponding to the M cell's location in the map is called its receptive field. The M cell has receptors for belief molecules, and it triggers the escape behavior when a sufficient number of belief molecules bind to its receptors. I don't want to get into mechanisms, but I want to point out that you don't need a neuron to do this. It's the kind of calculation that single-celled organisms and cells in your body do routinely. This figure shows the numerical Bayesian calculation that there is a predator in the receptive field compared to the same calculation done by counting how many belief molecules collide with the M cell over many trials. The model doesn't prove that placozoans actually do this, but it does show that it's possible for such simple animals to be nearly Bayes optimal decision makers in predator-prey encounters, and that they don't need a nervous system to do it. The model is so simple, requiring fairly minimal redeployment of existing mechanisms, that it would have been easy for natural selection to cobble this together when carnivores showed up in the mid-Ediacaran. The model works only because placozoans are tiny and flat. It fails on a larger scale because molecular diffusion scales badly. Once the map gets larger than a few hundred microns across, which is about the size of a large placozoan, diffusion is too slow for the belief to change quickly enough to keep up with changes in the world that it represents. I think it might not be a coincidence that our cerebrum and cerebellum comprise functional modules called cortical columns and microzones respectively that are each about as big as a medium to large size placozoan. Also, as far as I'm aware, there's only two places in the universe where you get epithelial tissues without tight junctions between the cells. That's in the ventral epithelium, the digestive epithelium of trichoplaques, and in brains, cortical tissues. That's necessary in this model because these belief molecules and the sensory molecules have to be able to flow around the cells in the epithelium without obstruction. To scale this mechanism up to something more than about 1 millimeter across, you'd need to evolve something like a system of tubes through which you could shoot particles across the map much faster than they could diffuse over those distances. This brings me back to the model of information transmission in vestibular sensory neurons that Larry Hoffman and I developed. The model's mathematically beautiful, but it's nuts. Why waste so much energy to transmit data that's mostly noise? Well, the placozoan model suggests an answer. The chemical brain mechanism needs particles representing samples from the statistical distribution of the raw data in the map. In the placozoan model, these particles are injected directly into the map. But the vertebrate vestibular apparatus is located millimeters or even centimeters away from the brain, depending on how big the animal is. It literally takes weeks for molecules to diffuse over those distances. To make this mechanism work in a larger animal, you're going to have to shoot those particles down some kind of tube so that they get into the map quickly enough. Instead of sending molecules along a tube from the sense organ into the brain, it might be even faster and cheaper to send an electrical pulse along a wire from the sense organ into the brain and have some mechanism at the other end of the wire that causes molecules to be released into the right part of the map. Our model shows that vestibular sensory neurons behave exactly as if that's what they're doing. The strangely complicated behavior of vestibular sensory neurons is a long-standing puzzle in vestibular neurophysiology, but the placozoan Bayesian brain model doesn't just solve this puzzle. It suggests that the vertebrate cerebellum is a Bayesian state estimator that uses essentially the same mechanism as in the placozoan particle filter model. It's just been upgraded using neurons to shunt particles around at high speed over much larger maps. So, I still haven't finished my PhD thesis. I don't have a model of how the cerebellum works. But now, I think I know where to start. As an added bonus, the Bayesian placozoan chemical brain model may be not just a foundation for modeling the cerebellum, it may provide a foundation for understanding general brain algorithms and mechanisms underlying perception and cognition. Before I wrap up, I just want to tell you one other fun fact about placozoans. I've suggested that placozoans or placozoan-like animals might be our ancestors. In fact, they might be our most recent ancestors that didn't have a nervous system. But placozoans reproduce by just pulling themselves in two. So that means that this placozoan that I found out at the Portobello Marine Lab last December might be an animal that's been pulling itself in two for more than 500 million years. If that's the case, then this might be not just a representative of a group of animals that used to be my ancestors. It might actually be my ancestor. And not just my ancestor, but your ancestor too. So it might be the great whatever grandmother of every creepy, crawly, slimy, fuzzy animal with a nervous system on Earth today. So I suggest that if you're lucky enough, as I have been, to meet a placozoan, you should treat it with enormous respect. I've told a story today about my research that cuts a straight path from sharks to placozoans as models of how the brain works. But of course that's not how it happened. I've lost count of the number of blind alleys and valleys of despair that I found myself in along the way. And I've lost count of all the people who've helped me. It's just a hopeless task to thank everybody by name. So I've just listed here my most recent cohort of graduate students who've contributed to the vestibular and placozoan models in the last few years. To everyone else, thank you. Your name might not be mentioned in this presentation, but I am grateful for your help and support. It's nearly 40 years since I accidentally became a zoologist after I slept through a few undergraduate zoology lectures and spent my days teaching myself how to code instead of showing up for class. When I first started as a programmer in the zoology department at Auckland University, I met people who spent their days studying things like the histology of sponges and the anatomy of sea urchin larvae. I remember thinking, these people are weirdos. Who cares about that stuff? I was interested in cool stuff like artificial intelligence and brains and consciousness. Why would anyone spend their days studying dumb animals like sponges and sea urchins? But I stayed in zoology and eventually turned into a weirdo myself, studying animals even dumber than sea urchins. I hope that after this talk you can see why I went down that path, but it took a long time to find it. And in the meantime, one thing that attracted me to and kept me in zoology was that zoologists are generally super excited not just about what they do, but about what other people around them are doing. Especially this department has had a supportive and vibrant research culture, and I'm grateful for the opportunity to have been a part of it for the last 30-odd years. I hope that continues and that today's young scientists can be as lucky as I've been. My advice to a young scientist would be never do anything because you think that some 20-something thinks it's cool, and be kind to weird old people. You might be a weird old person yourself someday, if you get lucky. And that's me done. Thank you. Here's a bonus for anyone who's stuck into the end of this video. In case you thought I was joking about going up to New Plymouth to catch up with the old gang at the WOMAD Festival, here is the old gang. We broke with tradition and shocked the authorities of our day by forming a soccer team and playing soccer instead of rugby. And just in case you're wondering, the serial killer is already locked up by this time.